So now let's go over to example 2. Got confused because I saw example 3 here, but it's because we've got 2 examples from pentose phosphate. So let's remind ourselves of the pentose phosphate. A little bit of context, right? So I gave you context of fermentation for the pyruvate decarboxylase. So now the context here is pentose phosphate pathway. This slide set is focusing mainly on the chemistry of these cofactors. However, we give it the context, and then the other slide sets, we expand a little bit more on each one of these pathways. So we know that the pentose phosphate pathway starts with glucose 6-phosphate. It goes through the oxidative phase, and then you can end up with ribose 5-phosphate to make nucleotides. That's what we focused on earlier on this semester, right? So the function of this is to make NADPH, to make fatty acids, to make a lot of other molecules, as well as to protect from oxidative stress. And then ribose 5-phosphate, which is used to make nucleotides. Okay. Now, if you need a lot of NADPH, right, you're pulling that strong to make molecules like fatty acids, but you don't need to make a bunch of nucleotides, well then there's no reason to go there. And so this pathway is very flexible. If we're looking at this right here, right, we see that strong requirement for NADPH, but not a need for high nucleotide synthesis, then instead of this going this way, we don't need that. So what will happen? These will go back to feed glycolysis. To feed glycolysis. So now we're expanding pentose phosphate pathway, right? If you need a lot of NADPH, but you really don't need much in the way of nucleotides. Now, again, as we've covered, it's not like 100% and 0%. There's always going to be push and pull depending on the control of the large negative delta G reactions and then the subsequent shift based on the substrates and products, right? But this just shows you that you can run this and then this and not make much in the way of nucleotides. Okay, so that's the context. Let's look at our example number 2 for TPP, transketolase. So the transketolase is this set of reactions right here for part of it. That is, when we look at this set of reactions right here, Zu5P, Zu5P, R5P, R5P, and then we combine them, and a whole bunch of stuff happens, and then we get fructose 6-phosphate and glyceraldehyde 3-phosphate. That's this, okay? And so we've got glyceraldehyde 3, fructose 6-phosphate, and fructose 6-phosphate coming out at the end. So the ones in beige are the ones that we end up with. So what the transketolase does, if you look at it, transketolase is involved in this reaction where you take these 2 molecules to produce these 2 molecules. And transketolase will take this molecule and this molecule and make these 2 molecules. Transketolase, so both of these, we've got some nice highlighting here. What it did is it took this 2-carbon fragment from this ketone and it added it to an aldehyde. See that? So it's like it transferred the ketone. Same thing when you look over from here there to here, instead of adding this to the ribose 5-phosphate, it's still, look at the other substrate here, it's an aldehyde. So it's going to take these and it's going to add it to that carbon. Boom. And when it adds it to the carbon, it converts that carbonyl group to a hydroxyl group. It converts that carbonyl group to a hydroxyl group. So how does this happen? How do we take a molecule, part of a molecule, and add it? Well, let's think about the previous TPP mechanism. Make it, break it, make it, break it. right? That is for carbon bonds, carbon-carbon bonds. So here we go. So let's remind ourselves where we've been. This is the enzyme mechanism that we just talked about. Okay, this is the pyruvate decarboxylase. So we're using this as a reference. Okay, we're looking for similarities and then we're looking for differences. So this particular diagram from the book shows the full TPP. So it's, again, I find it valuable to see multiple representations to remind ourselves, oh yeah, there's a lot more going on with the molecule itself, but you know, focusing in on the yelid. So the yelid, first of all, it got deprotonated. Let's not forget that. It got deprotonated and attacks the carbonyl group. Well, that's what that did over here. It attacked the carbonyl group. Okay. And that carbonyl group became a hydroxyl group. This carbonyl group became a hydroxyl group. Okay, so that looks very similar, right? You formed a carbon-carbon bond, and then the carbonyl that it attacked became a hydroxyl group. Same thing. Okay. Now, when we look at this, this is where, how does this look like this? Well, let's see, and does it, right? Well, first of all, if we go down here, we see that there is a resonance-stabled carbon ion, okay? Now, before that, there was no CO2 elimination. What we see is that, however, we made a bond, and then what we have is a base pulling off this hydroxyl group, electron rearrangement, and what we're doing is we're breaking that bond. We just broke that bond right there. So this is broke. This going there forms that aldehyde, right? So by pulling this off, you can go ahead and draw a base, pulling that off right now, right? As we do. We did it for over here, right? So why not? And then it breaks that bond, and then it forms this carbon ion. This carbon ion then attacks what? It attacks an aldehyde. Because remember, what is going on here? Well, we're going to, in this case, going down here, we're going to attach this to this, right? So what we've got is that, and what I mean by this is that an aldehyde. So we know an aldehyde's coming, right? So the carbon ion now attacks an aldehyde. And then what we have is a structure, if you look at this right here, that looks very similar to this up here, right? And so when you look at this and this, it's very similar, except now the chemistry, instead of pulling off a proton here and then breaking this bond, right, which is a carbon before the yelid, it breaks this one right here. So when we look at this, what we see is a make it, a break it, to release glyceraldehyde 3-phosphate, a make it, and then a break it. Those are the carbon-carbon bonds. Make it, break it, right? It's just in this case, it's not protonating the carbon ion, because its job is not to just protonate. Its job is to attack not a proton of an acid. Its job is to attack a carbonyl group to make another carbon-carbon bond to build a different molecule. So we're looking at the same thing right now. Again, we're looking at the Zu5P and the R5P. And so when we look at that, we're looking at the Zu5P and the R5P. We're looking at this reaction right there. Okay, so let's go through. Remember, for the first three, right, those are the same whether it's pyruvate decarboxylase or it is this transketolase reaction. So when you get to this step then, right, attack, kick it off, form a hydroxyl group, which is right there, electron rearrangement. And electron rearrangement, right, we can be a little bit better than that because we do know that this is base catalyzed. We know that there is a base pulling that off. Down to here. Okay, so now when we look at this, what is the leaving group? Right? For transketolase, the leaving group, this is what, is it the glyceraldehyde 3-phosphate? You think? No, it's the carbon ion. That's where those electrons went, right? So 3-carbon aldehyde is released, and the 2-carbon fragment now stays on as a reactive carbon ion. See, now think about this right now. For all of these, at this point, it depends on, right, whatever was added. What do we know happened? We know that whatever the carbonyl group was added, we know that then a bond had to have been broke. Make it, break it, right? So after you do that, you're left with something on there. Whatever that is, whether it's a 2-carbon fragment, whether there's some other enzyme out there that's got a 4-carbon fragment, what if it's got CH2, CH2, OPO3 2-? That's on there. That carbanion is ready to detect. Is it going to be a proton for a base? Is it going to be an aldehyde, right? If given different substrates, the idea for going through it like this is to be able to say, okay, I know the basic principles of these, and I know where things start deviating. But by knowing the fundamentals of it, ideally we can predict, right? But so now what we're looking at is we've got this reactive carbanion. So now this, again, you're going to have this resonance step right here all the time, right? And then we have this attacking the carbonyl group, again, forms an alcohol. And then this electron rearrangement, again, yeah, let's just draw a base pulling that proton off, right? Let's add to it. And then rearrangement. And then what do we have? Well, we could draw an arrow going back to, for the pyrophosphate, right? Because that regenerates the negatively charged ylide. And so what do we get? We pulled a 2-carbon fragment off of this 5-carbon molecule. Got it right there. After we pulled it off, the molecule was released as an aldehyde. Okay, so this molecule right here, right, was released as an aldehyde. And how did we get that aldehyde? This hydroxyl group right there was converted to an aldehyde. So when we look at this, the things to focus on are the similarities, the differences, how you can identify the make it, break it, make it, break it.